Hello everyone and welcome to our channel MooTechie. A very warm welcome to all the dairy technologists, students and food science enthusiasts joining us today. We're so glad to have you here as we dive into a fascinating and crucial topic. Let's begin our exploration of food microbiology with a special focus on the microbiology of milk and milk products. Milk and dairy products form a significant part of the human diet. They are wonderfully rich sources of essential nutrients like proteins, fats, vitamins and minerals. But here's the interesting part. Because they are so nutrient-rich, milk and milk products are also highly susceptible to rapid microbial growth. Now, in some wonderful instances, like when making yogurt or cheese, this microbial growth is actually beneficial and desired. But in other cases, it is completely undesirable and leads to spoilage. It's so important to remember that dairy products are vulnerable to spoilage or contamination with pathogens or microbial toxins. That's exactly why understanding the microbiology of milk is absolutely key for everyone in the dairy industry. Now let's look at the fascinating differences in milk from various animals. This table shows the average compositions in % of milk from different species. Looking at the constituents: water, fat, protein, lactose, ash, and total solids. For cow milk, water is 87.20%, fat is 3.70%, protein is 3.50%, lactose is 4.90%, ash is 0.70%, and total solids are 12.8%. For buffalo milk, water is 82.76%, fat is 7.38%, protein is 3.60%, lactose is 5.48%, ash is 0.78%, and total solids are 17.24%. For human milk, water is 87.43%, fat is 3.75%, protein is 1.63%, lactose is 6.98%, ash is 0.21%, and total solids are 12.57%. For goat milk, water is 86.5%, fat is 4.5%, protein is 3.5%, lactose is 4.7%, ash is 0.8%, and total solids are 13.5%. And for mairy milk, water is 89.1%, fat is 1.6%, protein is 2.7%, lactose is 6.1%, ash is 0.5%, and total solids are 10.90%. You can really see how the nutritional profile changes from one species to another. Moving on, we will see the wide variety of milk and milk products we encounter. These include raw milk, concentrated milk products like condensed milk, cream, butter, ghee and spreads, cheese and fermented milk, and ice cream and frozen desserts. Each of these has its own unique microbial story. Now, let's talk about a critical area: the sources of contamination. This is where things get really important for quality and safety. First, the animals themselves. The health of dairy animals is a very important parameter. This is because a number of serious diseases, including brucellosis, Q fever, salmonellosis, and staphylococcal and streptococcal infections may be transmitted to humans through milk. The microbes causing these diseases can get into the milk either directly from an infected udder, or indirectly through infected body discharges that may drop, splash, or even be blown into the milk. Next, the handlers. People involved in milking and handling can also be a source. Diseased persons may transmit illnesses like typhoid fever, scarlet fever, and diphtheria through contaminated hands or by coughing and sneezing during milking. And then we have the environment. The dairy farm environment can introduce pathogens at different stages. Some common airborne pathogens include streptococci, Corynebacterium diphtheriae, and Mycobacterium tuberculosis. Furthermore, contaminated water, fodder, and unclean vessels and containers can significantly contribute to pathogens and spoilage. Let us go further and examine more sources. It's fascinating to know that milk actually contains relatively few bacteria when it leaves the udder of a healthy cow. The problem often starts with contamination from the exterior of the udder and adjacent areas. Thankfully, this can be reduced by washing the udder and grooming the cow. The two main sources of contamination are dairy utensils and milk contact surfaces. To be specific, these sources include udder infection, dirty udders and teats, hands and udder cloths, and milking equipment. Modern dairy equipment is designed to help us. Modern dairy utensils, particularly milking machines and bulk milk coolers, are designed to provide easy access for cleaning, sanitizing, and drying. Excellent refrigeration is also a key control measure. But other possible contamination points are the hands and arms of the milker, the air of the barn, and even flies. The quality of the farm water is also crucial. And after the milk leaves the farm, we must consider the tanker truck, transfer pipes, sampling utensils, and the equipment at the processing plant. So what are the best practices? The udder should be cleaned before milking. The initial few strips of milk should be thrown out. Thumb milking should be avoided. And machine milking is generally better for hygienic milk production. Now let's look at other sources of contaminants. This is where details matter. Milking equipment and bulk storage tanks can make a significant contribution to the psychrotropic microflora of raw milk if they are not adequately sanitized. Exposure to inadequately cleaned equipment and contaminated air are also sources. Milk residues on surfaces can support the growth of many organisms. We find gram-negative organisms like Pseudomonas, Flavobacterium and Enterobacter aureus, and gram-positive organisms like Corynebacterium and spore-forming Bacillus and Clostridium. These organisms can be removed by effective cleaning, but they may build up as stubborn biofilms in poorly cleaned equipment. Another challenge is milkstone, a mineral deposit that can accumulate on inadequately cleaned surfaces, especially in hard water areas. Bacteria and spores can colonize this material and are then protected from cleaning and disinfection. Some may even survive pasteurization and eventually cause spoilage. It's a real hidden enemy. Now let's explore the concept of spoilage. The microbial quality of raw milk is absolutely crucial for producing quality dairy foods. Spoilage is a term used to describe the deterioration of a food's texture, color, odor or flavor to the point where it is unappetizing or unsuitable for human consumption. It often involves the degradation of protein, carbohydrates and fats by microorganisms or their enzymes. The defects that can occur in milk due to microbial growth are quite varied. They include 1. Off flavors, 2. Lipolysis with development of rancidity, 3. Gas production, 4. Fermentation to lactic acid with souring, 5. Coagulation of milk proteins, 6. And viscous or ropey texture and discoloration. It's amazing how many ways milk can spoil. We can classify these spoilage microorganisms by what they break down. Microorganisms degrading milk carbohydrate, which is lactose, are called glycolytes. Examples are Streptococci and Lactobacilli. Those degrading proteins are proteolytes, like Pseudomonas and Enterobacteriaceae. And those degrading lipids are lipolytes, such as Pseudomonas and Micrococci. Now let's look at a comprehensive table that links dairy products with their typical spoilage microorganisms. The table is titled Dairy Products and Typical Types of Spoilage Microorganisms or Microbial Activity. For raw milk, it's a wide variety of different microbes. For pasteurized milk, it's psychrotrophs, spore formers and microbial enzymatic degradation. For concentrated milk, it's spore-forming bacteria and osmophilic fungi. For dried milk, it's microbial enzymatic degradation. For butter, it's psychrotrophs and enzymatic degradation. For cultured buttermilk and sour cream, it's psychrotrophs, coliforms, yeasts and lactic acid bacteria. For cottage cheese, mauve, it's psychrotrophs, coliforms, yeasts, molds and microbial enzymatic degradation. For yogurt and yogurt-based drinks, it's yeasts. For other fermented dairy foods, it's fungi and coliforms. For cream cheese and processed cheese, it's fungi and spore-forming bacteria. For soft, fresh cheeses, it's psychrotrophs, coliforms, fungi, lactic acid bacteria and microbial enzymatic degradation. And for ripened cheeses, it's fungi, lactic acid bacteria, spore-forming bacteria, and microbial enzymatic degradation. This really shows how each product has its own specific microbial challenges. Here are the important genera of bacteria normally found in milk. It's quite a list: Microbacterium, Micrococcus, Streptococcus, Staphylococcus, Lactobacillus, Bacillus, Clostridium, Arthrobacter, Actinomyces, Coxiella, and Pseudomonas, among others. Some of these are particularly notorious. Species of Pseudomonas are prominent in producing defects in milk. They are associated with a bitter flavor and rancidity, as well as a fruity odor. That fruity odor is actually due to a mixture of ethyl butyrate and ethyl hexanoate. On the other hand, while lactic acid production is desirable in fermented products, it's undesirable in fresh milk. And a specific strain of Streptococcus lactis can produce a malty flavor defect in dairy products. Let's categorize the types of spoilage microorganisms. First, psychrotrophs. These represent a substantial percentage of the bacteria in raw milk, with pseudomonads being the predominant group. Importantly, 65 to 70% of the psychrotrophs isolated from raw milk are Pseudomonas species. Their key characteristics are their abilities to grow at low temperatures, between 3 and 7 degrees Celsius, and to hydrolyze and use large molecules of proteins and lipids for growth. They can also reduce the diacetyl content of buttermilk and sour cream, leading to an off flavor. And they are the bacteria that normally limit the shelf life of cottage cheese. Next, coliforms. Like psychrotrophs, they can also reduce diacetyl. In cheese production, if the starter cultures are slow, coliforms can grow quickly and produce gas. Then we have lactic acid bacteria. Sometimes, they can cause excessive viscosity in buttermilk and sour cream. They can also reduce diacetyl-EDAC, resulting in a yogurt-like flavor. And heterofermentative types can develop off flavors and gas in ripened cheeses. Now, let's talk about fungi. Yeasts can grow well at the low pH of cultured products like buttermilk and sour cream, producing off flavors described as fermented or yeasty. They are a major cause of spoilage in yogurt. Molds can grow on the surfaces of cheeses when oxygen is present. In packaged cheeses, mold growth is limited by oxygen availability, but some, like penicillium, can still grow. Cream cheeses are susceptible to spoilage by heat-resistant molds, like Bisoclamys nivea. Now on to spore-forming bacteria. Raw milk is the usual source of these in finished products. Common ones are Bacillus licheniformis and B. cereus. They can produce enzymes that coagulate milk protein. In aged cheeses, if proteolysis occurs, it can favor the growth of Clostridia adsat, which produces gas and butyric acid. Occasionally, gassy defects in processed cheeses are caused by Clostridium butyricum. Let's look at a table summarizing the causes of gassiness in different types of cheese. The table lists the organism and the cheese affected. Coliforms cause early blowing in raw milk pasta filata cheese. Yeasts cause early blowing in Domiati and Camembert. Lactobacillus fermentum causes late blowing in provolone. Heterofermentative lactobacilli cause late blowing in cheddar and Gouda. Propionibacteria cause late blowing in Schrin's cheese. Clostridium tyrobutyricum causes late blowing in Gouda and Swiss cheese. And Eubacterium species cause late blowing in cheddar cheese. It's so specific. Thermoduric bacteria are common causes of spoilage in concentrated milks. Other microorganisms like Eubacterium sera can also cause gassiness. There's even a rare white spot defect in Swiss cheese caused by an enterococcus. Now, a fascinating indirect cause of spoilage, enzymatic degradation. Microbial enzymes like proteases and lipases can remain active even after the microbes that produce them are destroyed. Populations of psychrotrophs can produce enough extracellular enzymes to cause detectable defects. These heat-stable enzymes have even been known to spoil UHT milk. Pseudomonas fluorescens is a common producer of these troublesome lipases. Let's get into specific product defects, starting with butter. Butter is primarily fat. Its main defect is rancidity, caused either by autooxidation or by microbial lipases. The presence of short-chain acids like butyric acid results in that unmistakable rancid flavor. Molds can also grow on the surface. Putrid and fruity flavors are caused by psychrotrophic bacteria. It's so important to remember that if the cream used to make the butter has a defect, it will carry through to the final product. Now let's consider cheese. Defects in the original milk can appear in the cheese made from it. In cottage cheese, you might see gelatinous curd formation. Lipolytic and proteolytic organisms cause defects, and bacteria like P. fragi can cause a slimy curd. You might also encounter fruity, putrid, or rancid odors. E. coli can cause unclean flavors and even a gassy defect. Molds multiply slowly, but growth of penicillium or mucor can appear on the surface. Some of these molds can even produce mycotoxins, which is a serious health concern. Here is a table listing some common defects in fluid milk and their associated microorganisms. A bitter flavor is associated with psychrotrophic bacteria and bacillus. A rancidity flavor with psychrotrophic bacteria. A fruity flavor with psychrotrophic bacteria. Coagulation with bacillus species. A sour flavor with lactic acid bacteria. A malty flavor with lactic acid bacteria. And a ropey texture with lactic acid bacteria. Now let's look at defects in cream. The table lists souring caused by uncontrolled acid development from lactic acid bacteria. Sweet curdling caused by Bacillus cereus producing milk clotting enzymes. Bitterness caused by protein degradation from Pseudomonas and Bacillus subtilis. Microbial rancidity caused by bacteria like Pseudomonas fluorescens and yeasts. Fruity flavor caused by paint P.S. fragi. Yeasty flavor caused by lactose fermenting yeasts. And silmy or ropey cream caused by Alcaligenes viscolactis. Following this we have defects in butter. The table lists microbial defects in color and flavor. Black discoloration caused by Pseudomonas nigrifaciens. Moldy butter caused by various molds. Orange and yellow discoloration caused by Geotrichum candidum. Rancid flavor caused by lipase action from Pseudomonas and yeasts. Putrefactive taint caused by protein breakdown. And a fishy taint caused by action on lecithin. This is a lot of information, so let's summarize the significance of different bacteria and how to control them. We have a table for that. For lactic acid bacteria, which cause souring, we control them by pasteurization and low temperature storage. For coliforms, which produce gas and unclean flavors, we use pasteurization and strict hygiene. For psychrotrophs, which cause off flavors and produce heat-stable enzymes, we use dekinimization, pasteurization and hygiene. And for heat-resistant bacteria which spoil heat-treated milk, clean milk production is key to reducing their initial numbers. Now, we move to a very serious topic: diseases transmitted through milk. The diseases we'll cover are 1. Tuberculosis, 2. Brucellosis, 3. Diphtheria, 4. Q fever, 5. Viral infections, 6. And listeriosis. It's important to know that the following bacterial pathogens are still of concern today in raw milk: Bacillus cereus, Listeria monocytogenes, Salmonella species, and E. coli O157, H7, among others. Let's discuss each one. Starting with tuberculosis. It can be caused by Mycobacterium tuberculosis from humans or Mycobacterium bovis from cattle. The human type can even get into milk from infected handlers. It's sneaky because an animal might test negative initially, but positive after a few months. Symptoms include pulmonary infiltration, cough, fever, and weight loss, with an incubation period of 4 to 6 weeks. Prevention and control are vital. Animals should be tuberculin tested. Infected animals should be isolated. Proper heat treatment of milk is essential, and the traditional habit of boiling milk in India is excellent for preventing this. Overcrowding must be avoided, and tuberculosis patients should not handle milk. Next, brucellosis. This is one of the most common milk-borne diseases. It's a highly contagious zoonosis caused by Brucella species, which are small, gram-negative rods. Symptoms include profuse sweating and joint and muscle pain. To prevent it, we must segregate or slaughter infected herds, vaccinate herds properly, and ensure adequate heat treatment of milk. Now, diphtheria. This is caused by toxigenic strains of Corynebacterium diphtheriae. It's an upper respiratory tract infection characterized by a sore throat and an adherent pseudomembrane. Prevention involves adequate heat treatment of milk, preventing infected persons from handling milk, avoiding unhygienic practices by workers, and proper vaccination. Now let's talk about Q fever. This is caused by Coxiella burnetii, which is notably more heat-resistant than even tuberculosis. It can survive inadequate pasteurization and freezing. It's viable for a long time and remains viable in some cheeses for many days, though yogurt's acidity kills it within 24 hours. Human infection often comes from inhaling infected dust. Symptoms include high fever, headache, and pneumonia. Prevention involves adequate heating of milk, separating calving sheds from milking areas, vaccinating animals' bays, and conducting prevalence surveys. We also have salmonellosis, which includes typhoid and paratyphoid fevers. Sources of contamination include the hands of carriers, polluted water, and flies. Then there's poliomyelitis, caused by polioviruses. It's an acute viral disease spread primarily via the fecal-oral route. Thankfully, we have two effective vaccines, the inactivated Salk vaccine and the live oral Sabin vaccine, which have been crucial in controlling the disease worldwide. Infectious hepatitis, caused by hepatitis A virus, is also spread by the fecal-oral route, often in conditions of poor sanitation. The incubation period is between 2 and 6 weeks. A key symptom is jaundice, a yellowing of the skin and eyes. Finally, we have listeriosis caused by Listeria monocytogenes. This is a particularly concerning and versatile pathogen. It's ubiquitous in the environment and can be found in raw milk, pasteurized milk, cheeses, and many other foods. Its terrifying ability to grow at temperatures as low as 0 degrees Celsius allows it to multiply in refrigerated foods. It's one of the most virulent foodborne pathogens, with a high mortality rate in clinical infections. And with that, we have reached the end of our detailed journey into the microbiology of milk and milk products. Thank you so much for joining us and for your interest in this vital field of food science. We hope this session was insightful and will help you in your work or studies. Thank you. See you in the next class.